The interdisciplinary meeting of antimicrobial resistance and innovation called IMARI has launched. And the first version of IMARI took place in Las Vegas a few weeks ago and brought together researchers, clinicians, industry leaders, and policymakers to address one of the greatest challenges in modern medicine, antimicrobial resistance. This inaugural conference was the first time that AASM, the American Society for Microbiology, and the Infectious Disease Society of America collaborate together in a scientific meeting in a long time. One of the sessions involved an unprecedented collaboration between the two journals published for the different societies, Antimicrobial Agents and Chemotherapy, published by the ASM, and the Journal of Infectious Diseases, published by the Infectious Disease Society of America. To celebrate this collaboration, we had a session at IMARI called the Best AMR Antimicrobial Resistant Papers of 2025 for AAC and JID. And this is a recording from not live, but a podcast that we prepared for this session. And to discuss these important papers, we have two really outstanding, upcoming young investigators. Dr. Aisha Khan, who is Assistant Professor of Pathology and Laboratory Medicine and Associate Director of the Division of Clinical Microbiology, Biomedical and Translational Research Track, and the Principal Investigator of the UC3P Prime Pre-Health Pathway Program, and Dr. Madison Stelfox, who is Assistant Professor of Medicine at the University of Pittsburgh Medical Center in Pittsburgh, Philadelphia. So welcome to the show and welcome to Editors in Conversations. The objectives of the podcast is to review the best papers on topics of AMR in 2025, publishing the top journals of ASM, which is Antimicrobial Agents and Chemotherapy, and the Journal of Infectious Diseases from IDSA. Discuss the context of each finding and contribution to the AMR community, and elaborate on the implications of antimicrobial resistance, the novelty in the field, and how the field is advancing with these publications. This episode is brought to you by the Antimicrobial Agents and Chemotherapy Journal, available at asm.org/AAC. I'm your host, Cesar Arias. I'm the editor-in-chief of AAC, and this podcast is supported by the American Society for Microbiology, which publishes AAC. If you plan to publish in AAC, ASM members get up to 50% off on publishing fees, and visit asm.org/membership to sign up. And don't forget to check the latest issue of AAC available at asm.org/AAC with outstanding papers on mechanisms of resistance, pharmacology, antimicrobial agents, epidemiology, and clinical therapeutics, among others. IDSA through JID also launched a section of antimicrobial resistant papers recently, and we have also great papers in JID that we're going to highlight. Without further ado, we're going to start this exciting session with the first paper. Dr. Khan, tell us about the first paper and why you chose those.
So the first paper that I chose was one of the most highly cited papers, but I think the findings really explain it. So it's called a microbiological and structural analysis of the interplay between sulbactam, durlobactam, and imipenem against PVPs of Acinetobacter. So the reason I chose this is because I think it applies a strategy that we're very familiar with when it comes to the treatment of very severe, recalcitrant, difficult to treat gram-positive pathogens, target redundancy. So the ability to use two agents that are guided by a mechanistic rationale because they essentially saturate multiple complementary penicillin binding proteins, and applying that strategy to gram-negatives, which is increasingly popular in the realm of carbapenem-resistant organisms. So the paper essentially is it's an aftermath of the original ATTACK clinical trial, where incidentally, because of a lot of carbapenem-resistant Acinetobacter infections being so difficult to treat and also oftentimes being polymicrobial in very complex patients, imipenem was included in both arms, so both the sulbactam durlobactam and the comparator colistin arms. And even though, as a result, sulbactam durlobactam was clearly seen to be a very beneficial antibiotic for the treatment of carbapenem-resistant Acinetobacter, and it's now recommended by IDSA as the primary go-to treatment if available. I think it's cool that the study essentially showed using essentially time kill assays, pretty simple techniques that we've been using for the last 50, 100 years, time kill assays, and something new, molecular docking analysis, which is essentially simulation and modeling, to show that two different agents can bind two different PVPs. So in this case, sulbactam, which binds PVP3 really well, and then imipenem, which binds PVP2 really well. And they come together and display synergy in a way where there's more bactericidal killing than any of the agents alone. So clear benefit to potentially, and supports IDSA's recommendation in combining sulbactam durlobactam with a carbapenem for the treatment of CRAB.
Very interesting concept that we knew about this in gram-positive bacteria for a long time, of what we call PVP saturation and differential PVP, that now is very hot. We actually had a very amazing session in IMARI about this concept and how you can use, even if the organism is reported as resistant to the particular carbapenem, the addition of the two beta-lactams to different PVPs can produce synergistic effects and probably explain some of the clinical findings. So you see, the gram-negative still can learn a lot from the gram-positives, which is I'm very glad to hear. Okay, Dr. Stelfox, how about your paper?
So this is actually a great transition to the paper that I chose in JID, which was titled Ampicillin sulbactam in combination with ceftazidime avibactam against metallo-beta-lactamase producing carbapenem-resistant Acinetobacter baumannii in a genomics-informed mechanism-based model. So this was published online in November of 2025 in JID, and for similar reasons is why I chose this paper. Carbapenem-resistant Acinetobacter baumannii is a significant challenge, and we need to really find the best ways we can treat these difficult infections. And I really liked how this paper looked at this idea from a pharmacokinetic modeling aspect, and really kind of delved into developing a really sophisticated way of looking at this and helping us understand why these combinations can be effective, even though you maybe not think that they would be for these isolates. So they developed a model to predict the bacterial cytoactivity of combining ampicillin sulbactam and ceftazidime avibactam in Acinetobacter baumannii isolates that either harbored metallo-beta-lactamases or class D serine beta-lactamases. And then they combined traditional PK/PD parameters for these antibiotics, like serum-free concentrations, half-life, etc., with additional modeling for small molecule diffusion, so how these beta-lactams get into the cell, or the bacterial cell, and then also using enzyme kinetics for how the beta-lactamases are functioning, efflux pouring content that they all found with whole genome sequencing of these isolates, and then kind of pulled the whole model together and validated it in an in vitro one compartment model to look at, do we think we know the mechanism of why this combination works? Do we see our model recapitulating that? And I think the major findings are really summarized in figure 3, where they show you all of the parameters they assess for this model. You can see the enzyme kinetics they're using, and I think the graphical representation of it is really helpful. And then in figure 5, where they put their in silico predictions of the model and layer it over the actual bacterial killing they see in their in vitro one compartment model to show the efficacy of the combination even in these highly drug resistant bacteria. And a lot of what they talk about is the idea of hitting all of these different PVPs, getting that PVP2 with the avibactam in addition to everything else to really get that synergistic efficacy there. So That's why I picked this paper, kind of dovetailed really well with that first paper, Aisha.
Yeah, and of course, here the element is the presence of the enzyme or beta lactamases, which add a
Mm-hmm.
little bit more complexity to the whole thing. But
Correct.
highlights in particular the understanding of the molecular mechanisms is absolutely crucial to really think about how you deploy your drugs. Right.
Mm-hmm.
So,
Exactly.
for all the infectious disease doctors that are listening to it, we really need to dig deeper into these mechanisms to try to get how to treat these organisms that are sometimes so difficult to treat. Excellent. Excellent. And very similar topic. So, you read both papers, you really learn a lot. Aisha, which one is the next one?
So it's a little related to learning more about mechanisms to guide treatment. So very recently, over the last 10 years, I think clinicians are getting more used to understanding types of enzymes and beta lactamases to a very granular level, because our treatment recommendations, like the IDSA treatment guidelines for CRE, are directly based on mechanistic understanding of what you can use for serine carbonamases versus, even within serine carbonamases, KPCs versus OXA48. So this paper is focusing on OXA48, and it's titled Advancements in the Fight Against Globally Distributed OXA48 Carbapenemase, Evaluating the New Generation of Carbapenemase Inhibitors. So it's looking at a lot of the inhibitors that are either recently introduced and clinically available or in the pipeline, so soon to be available, and broadly in two classes, right? So it's looking at diazobicyclooctanes, which are these novel, interesting beta lactamase inhibitors that also have anti-PBP2 activity in Enterobacterialis. So they have standalone antibacterial activity, though when they're paired, they can be far more potent. And then it's also looking at a new class of bicyclic boronates, which are metallo-beta lactamase inhibitors, which are, again, that's unprecedented. So all of our beta lactamase inhibitor development so far is really focused on serine beta lactamases. So an example of a metallo beta lactamase inhibitor is zuroburbactam, so something that is, again, in the pipeline and soon to be available, hopefully. But it's really evaluating the utility of these beta lactamase inhibitors or potentiators, if they're going beyond just inhibiting beta lactamases, against OXA48. And the study essentially does kinetic assays, susceptibility testing of a small panel of clinical isolates, as well as isogenic strains that are expressing OXA48 and ESBLs like CTXM15. And molecular modeling to show collectively that the most potent by far are Zarubobactam, Durlobactam, and something that we already have available, Avibactam. And I thought it was more interesting that Avibactam was so comparable in a lot of these assays to the level of potency and efficiency that Zarubobactam and Durlobactam showed. So I think it also shows that in addition to the new things coming in the pipeline, we need to actually preserve what we already have. I know there's a lot of institutions that use a lot of ceftazidime avibactam, and even though in the global north and America we don't have a lot of OXA48s, I think we've learned our lesson with NDMs that maybe we should use antimicrobial stewardship and think very carefully about what we're using. So I think this really, hit that point home.
That is a very interesting situation now, where choosing the right beta lactamase inhibitor is becoming a real conundrum for a lot of us clinicians, um, and is beyond the common knowledge that is out there, and particularly and OXA48 much more prevalent in Europe and some part of North Africa than in the United States. But we start seeing like the metallo strains now coming to the United States and understanding this, and the interplay between the beta lactamase inhibition and the PVP inhibition done by the two molecules, and that creates really headaches for people like in the CLSI to establish breakpoints and determine exactly where to draw the line. So this is from the scientific point of view is incredibly exciting. But from the clinical point of view, are options that we hadn't thought before, and now we start having more data that will help us just move the needle forward and save lives basically, because using these drugs against these organisms can make a huge difference. Beautiful. So Madison, how about you? What's your next one?
All right. So this is shifting gears a little bit into the gram-positive world.
Yay!
And this article was published online in November 2025, and it is titled "Daptomycin-Loaded Nanocarriers Facilitate Synergistic Killing of Methicillin-Resistant Staph aureus via Lipid-Mediated Interactions and Targeting." And so the reason I chose this paper is I think we all know that our antibiotic discovery pipeline is challenging. And so novel ways to improve the efficacy of already approved drugs is an important way that we can continue to build up our antimicrobial toolkit. And then MRSA remains a significant clinical challenge. I think that Maxim still holds that infectious disease fellowship is essentially a Staph aureus fellowship. And so any treatments that can help us combat these challenging infections will be helpful. So the main topic of this paper, the authors were looking to build lipid-based nanoparticles that they call cubosomes because of the cuboid shape that they eventually form. And can they load them with the membrane-targeting antibiotic daptomycin to enhance delivery to the bacterial cell surface and improve efficacy? And so the major findings of this paper were they were able to build these cubosomes with, and I hope I'm pronouncing this correctly, but it's phytantriol, which is an aliphatic alcohol, and then load it with daptomycin in kind of inside the cuboid structure. And so that it was stably in there, it was able to leach back out in an efficient manner. And then they looked at daptomycin, the cubosomes, and then together to see how the efficacy worked both in vitro and in vivo. So they saw greater bactericidal activity of the daptomycin cubosome combination against 14 MRSA isolates using the minimum bactericidal concentration as their assay. And then they also did something called neutron reflectivity, which I had to look up. But essentially, they built like man-made MRSA lipid bilayers using the lipid content of the Staph aureus, and then used this technology to kind of measure the thickness and the lipid content of these artificial membranes after treatment. And so they saw that when you combine the daptomycin and the cubosomes, you're actually able to disrupt the membrane even more than either alone. And then they also did an in vivo test. They did a murine bacteremia model where they compared daptomycin plus daptomycin and the cubosomes to look at, did they see better bactericidal activity in the mouse model as well. And this is a very quick mouse model. It was like a 24-hour experiment. But they saw that they did get better killing of the Staph aureus in that model with the cubosomes. So I thought it was a really interesting way to kind of look at a old antibiotic in a new way, and I just thought that was a great way to kind of highlight some novel ideas in antimicrobial.
Absolutely, it's very interesting how these new, particularly nanotechnology platforms, are helping with the delivery, and particularly these big drugs like daptomycin. I know there has been a lot of work with amphotericin B to, for example, decrease the amount of amphotericin B and therefore toxicity on this, and then have a much more targeted delivery. And this is a very interesting area that definitely there's some compounds now in the pipeline that these compounds help that. And in other areas like HIV, for example, also devices specifically designed to do this release and slow release of these drugs that have a very seem to have very good activity. But it's very nice that in this paper there was even in vivo proof of concept that this is a viable platform to move forward. So it's great to see.
Exactly.
Excellent. Let's go to the next one from AAC. I hope you tell us something about gram-positive, Dr. Ken.
Yeah, and I'm going to actually start with streptococcus because I feel like streptococcus are always forgotten in the realm of antimicrobial resistance sometimes, or not as focused on. And to me, at least, it's interesting because I focus a lot more on enterococci and Staph aureus. So this paper is titled Amoxicillin Non-Susceptible Strep Pneumo Causing Invasive Pneumonia, and it's a dissection of, I think it's fascinating because it actually connects the dots between antimicrobial resistance, virulence, and also does a good job of connecting the dots between in vitro and clinical outcomes. So I thought it was actually a very beautiful story. It's all in one hospital in Barcelona in Spain, and they essentially look at strep pneumo isolates that were collected at their institution, about 1,700 or so isolates between 1994 and 2020. And they do, again, use genomics to their advantage to do a dissection of susceptibility, along with understanding the impact of serotype and understanding how vaccines change the landscape, epidemiological landscape of susceptibility. So, I mean, good news, amoxicillin susceptibility overall increased from about 75% to 91% between 1990, early 1990s and 2020, probably due to the impact of the vaccine. But not so great news. That was very specific to a smaller subset of low-level resistance. It looks like the proportion of isolates that were really highly resistant, so those with MICs greater than 2, remained stable across the years, so about 5% prevalence. So it looks like what vaccine pressure, selective pressure, did from vaccines is that the most resistance clones essentially survived over time. And they also go on to do a more detailed dissection to show that specifically it's one resistant lineage that's kind of dominated over the years, GPSC6. And then they focus on, PBP mutations. And I think it's interesting because they actually showed there's a stepwise progression to really high level resistance, that for low level resistance, you could have mutations in PP2B and 2X. But to get really high level resistance, it's like this additive layering of PBP mutations that leads to essentially the worst outcomes, because the end of the paper, I think, does a good job of connecting the dots between outcomes and shows essentially that there's not too many differences in mortality unless you look at the group of patients that are treated with amoxicillin that have higher than MICs higher than 2. That seem to have a clear increased mortality with amoxicillin therapy. And I mean, lastly, I think one of the most interesting parts of this paper was the commentary that Dr. Dan Musher wrote, speaking to this specific point, because I think a lot of the conclusions of the paper were that amoxicillin is worse than third-generation cephalosporins like ceftriaxone. And I think the commentary is a good read because it really goes into how studies like this do need to account for differences in dosage, route, times of administration, because his whole commentary focused on how that may have accounted for the differences in mortality, that it might not truly be that amoxicillin is in fact worse than ceftriaxone, since drug levels can be so variable when you're not accounting for, and the paper didn't fully account for the differences within the administration realm of everything related to probably what Madison can talk a lot more about, which is PKPD.
Yeah, absolutely. I actually serve as editor of this paper and commissioned that commentary because, of course, a penicillin resistant in pneumococci is something that is important, but particularly all the way to cephalosporin resistant is something that could be a very important development, and particularly in Spain, there seems to be appearing clones that seems to have reduced susceptibility to beta-lactams all the way from the penicillin to the cephalosporins. So this is one of the largest data sets that I remember that at least put that into context of clinical outcomes. But as any clinical outcome in retrospective, there are so many limitations and caveats. But it's a very provoking thinking about when this could happen, and if there is any reason to change the current practices to start with pneumococcus. Pneumonia is one of the most common infections in the world, and amoxicillin is one of the cheapest antibiotics, and accessibility of that antibiotic is very important, and how to optimize the use of these antibiotics is crucial. So great paper, and great choice. Madison, next.
All right. So this is changing kingdoms and talking about fungi. So this paper was published online in September 2025, and it's entitled Blood Cultures Contain Populations of Genetically Diverse Candida albicans strains that may differ in echinocandin tolerance and fitness. And so I had about 3 reasons why I chose this paper. One, it was an AMR paper that talked about antimicrobial tolerance in fungi. So I think that that's a really important thing to discuss clinically. And then it also looked at assessing infections as complex populations of microbes rather than clonal strains. So I think it's a really important concept to discuss. And then that kind of dovetailed into the ideas of antimicrobial tolerance and heteroresistance in that AMR discussion. So the main question of this paper was looking at within host genetic and phenotypic diversity of Candida albicans during fungemia in patients. And so there were 4 patients who had 10 colonies/strains chosen for whole genome sequencing to look at the within host genetic differences. And they found SNPs and indels, but they also found these large chromosomal changes in aneuploidy. And then they really drilled down into one particular patient's isolates, and they looked at the 2 main genetic subpopulations in that patient's Candida albicans isolates, mainly those that are euploid, and then those that were had trisomy of chromosome 7, which they call tri7. And they look at these subpopulations both in vitro and in vivo. And so interestingly, the euploid subpopulation and the tri7 subpopulation had the same MIC to mycofungin. And in media growth without any antibiotics, they grew similarly, so no real fitness cost. However, when they did competition assays, so mixing them 50-50, and then treating with mycofungin, that really selected for the euploid cells. And they did some antimicrobial tolerance studies to show that those euploid cells were more tolerant to the mycofungin. And then they also looked at more like virulence and colonization effects. And in those cases, when they did these competitive competition assays, the tri7 really kind of came out to be more fit. So it grew more in blood. The tri7s, they colonized the GI tract more. They, in a disseminated candidiasis model, it was definitely the more prevalent of the 2. However, as soon as you add mycofungin, that flips. And now you're really selecting for the euploid subpopulation. And then looking at this patient longitudinally, they saw the same thing happening in like the clinic, where as soon as the patient is on mycofungin, the population of the tri7s go down and it's more the euploid. So kind of looking at those population dynamics over the course of infection and how that can both be selective to help the infection get started and then help the infection stay infecting during antimicrobial treatment, I thought was really interesting, and really capitulated things we are seeing in bacteria and also in Canada as well.
And were those infections mostly bloodstream infections, or were of the other kind?
For the 4 patients they looked at, they were all bloodstream infections. One was immunocompromised. I believe the other 3 patients were not. I think 2 of them were endovascular infections.
Yeah. And it's interesting that you don't think about Candida being as flexible as bacteria because of the eukaryotic nature of their genomes, but they are incredible. Fungi are hot right now, and we're learning more and more about the genomic plasticity, which that doesn't seem to amaze. And hopefully, with the new antifungals in the pipeline, we will have more options. But we're learning more and more with the current ones that we are having. So this is absolutely interesting and very clinically relevant in the kind of patients that we're having to treat right now. Excellent. So fungi, Aisha, anything similar?
Yeah, so I chose the paper titled Anti-Biofilm Activity of a bunch of the new antifungals, Minogapix, iberxafungerp, rezafungin, et cetera, and comparing it to things, last line agents that we already know, like amphotericin B, specifically against Candida biofilms. So I thought this paper was really interesting. We already know, it's well known the biofilms are hard to treat, but I think I like that the paper methodologically tried to take what we do in terms of MIC testing and translate that to, how do we really test for susceptibility of a biofilm, a complicated, complex community of microbes to specific agents. So they essentially, that's exactly what they did. So they tried to understand if they build layers of biofilms and then throw the same agents at them and compare efficacy to planktonic cells, how do they do? And I think the findings were pretty interesting. First, as predicted, agents that are normally are able to kill planktonic cells anywhere between 0.5 to 2 micrograms per mil, require up to 8,000, 10,000 micrograms per mil to eradicate biofilms across species. So I think that we can, that's predictable, largely due to the nature of, the more complex dormant cells that are buried within the biofilm that are really hard to reach. But something interesting was Minogapix, which is a newer antifungal, displayed pretty potent, the most superior activity across species. Only about 3 to 10-fold more was needed to eradicate a biofilm compared to the planktonic version of the cells, with the exception of Candida auris, where iberxafungerp was better. So I think it also went into understanding species and clades and how that matters, really getting down to understanding mechanisms of action between as the newer antifungals are rolling out. Amphotericin B was interesting because even though it wasn't the most potent, it had the smallest MIC changes. So the difference between MICs of planktonic cells and the biofilms were the smallest. So there was a mechanistic clue there, since amphotericin B targets membranes, that maybe biofilms have a difficult time coping with perturbations of membranes as opposed to like echinocandins, which they're much easier, have an easier time facing off against. And caspofungin was really not great, had the worst activity against biofilms, and that's concerning because there's clinical implications to the widespread use of caspofungin, including to treat infections where we imagine there to be a higher burden, sticky infections, biofilm-forming infections. So I think the paper had, very interesting implications in just anyone across fungi, bacteria, trying to understand and compare the efficacy of any antimicrobial agent within a biofilm community versus a planktonic community.
Yeah, this is these assays are very complicated to do and difficult to interpret sometimes. So kudos to be so rigorous with this analysis. And this is something that we talk about MICs as a way to treat patients in a very static in vitro system, and this is sometimes the translational effects in real life are not that critical. Biofilm eventually is something that we can relate more, particularly in catheter-associated infection and other infections, specifically with some of these Candida species. And this is hoping that in the pipeline, unfortunately, some of these drugs have been dropped from development, but the others continue, that we will have some areas where we can use them against this type of infections. Did they comment on combinations of any of these drugs? Because that's another area where people are trying in terms of biofilm activity.
Yeah, they didn't go too much into combinations. I think it was so complicated to even compare. They, in addition to MICs, they did things called, MBCs, minimum biofilm eradication, minimum biofilm damaging concentration. I think they had to combine so many different assays to show that this is real. That I think it would, but the next level I think is trying to see how do you do in general synergy testing, right? We're still doing single drug, single bug in the lab to be able to guide organisms that are almost exclusively treated with combinations, right? Like Acinetobacter. So I think in general, development of more feasible synergy testing is critical.
Absolutely. So, and this is an area of evolving, and the clinical implications of this are still, unknown really about how we can translate this. But, this is a good start to start understanding how these drugs can potentially help us treating these very difficult infections, particularly Candida auris, which has become a real public health problem. Okay, let's move to the fourth paper from JID.
All right. So this is now in the mycobacterial world. And this paper was published in the April 2025 volume of JID, and it's entitled Engineered Mycobacteriophage, TM4, Rapidly Determines Bedaquiline, Protominid, Linazolid, Rifampicin, and Clofazimine Sensitivity in Mycobacterium tuberculosis clinical isolates.
And of course, you love phages, don't you?
I did, but this is not looking at them from a therapeutical standpoint. This is looking at them as a biological sensor. And so I chose this paper because it really focuses on MDR tuberculosis, which is a huge public health concern and clinically challenging to manage. I think part of that difficulty is how long it can take to get reliable antimicrobial resistance testing for these slow-growing isolates. And so these authors looked to build a phage infection-based phenotypic antimicrobial resistance detection system, building on some prior work to build it faster, more sensitive, more stable, and less labor-intensive. So they took the broad host range mycobacteriophage TM4 and genetically engineered it to contain something called green enhanced nanolantern, which is where you take M nanogreen fluorophore connected to nanoluciferase to, I think it's one of the brightest fluorescent probes that you have. And then use that as a detection for viable Mycobacterium tuberculosis. So as far as the authors know, all clinical strains are susceptible to this phage. And to get phage infection and transcription, you have to have viable bacteria. So if they couple the phage with inhibitory concentrations of anti-tuberculosis drugs and they don't see any fluorescence, that means there's no live bacteria there. But if they do see fluorescence above the threshold that they determine, then those bacteria are living and surviving in that antibiotic concentration. And so they were able to look at essentially the reporter for all of these various tuberculosis drugs and then compared it to a subset of isolates that they collected all over the world, Africa, North America, and I think South America as well, to kind of sample as much of the broad scope of tuberculosis as they can. And then they compared them to the gold standard for the phenotypic assays for antimicrobial susceptibility and saw excellent agreement. And they also did this in two different labs. So they said, okay, we can do this and we see the results. And then they also recapitulated the results in a subset in another lab. And so I thought that was just a really interesting way to look at antimicrobial susceptibility, especially in these newer drugs where, genomic sequencing may not be able to predict whether this is sensitive or resistant, either because it's complex or we don't really know how resistance behaves, or there's multiple ways to become resistant. And the fact that this only needs a plate reader and a 96-well plate is also a great technological advance. So I thought it was an interesting way to use phages, not for treatment, but for assessing antimicrobial resistance.
This is a pretty cool way to use a phage, huh?
Yeah.
That is, and this is so important because diagnosis of multi-drug resistance in these organisms completely changed the course of diagnosis. I remember, there have been a lot of efforts to use CRISPR-Cas, for example, sort of with a similar rationale to try to detect subpopulations of drugs that have specific mutations, for example, for rifampin resistance and other in Mycobacterium. But this is a very ingenious way to use, a biological sort of dynamics to really get susceptibility testing, which is something that we wait for phenotypic characterization for too long now. Now whole genome sequencing are helping, but it's still a week away. And with this, you could potentially have signals very, very quickly. And this, so pretty cool paper.
They said
Thank you very
48
much.
hours. 24 hours to grow, and then 24 hours to do
That
the
is amazing.
separase
That
assay.
is pretty cool. Excellent. Okay, Aisha, your last paper of the session that you choose for 2025 in AAC. Tell us about it.
Okay. I actually think this is a really interesting paper because it has implications across organism groups, bacteria, fungi, and it comes from TB. So that's a little rare. Usually we take what we know in bacteria and translate it to everything else. The paper is called Emergence of Antibiotic-Specific Mycobacterium Tuberculosis Phenotypes During Prolonged Treatment of Mice. So it's really getting at this phenomenon, which I think is a very hot topic now, which is, there isn't a binary of susceptible and resistant, but then there's all of these other phenotypes like tolerance and heteroresistance, and they're specifically talking about persistence. Which historically has been thought of as a dormant, metabolically inactive state, where there's a subset, a much smaller population, subpopulation of cells that are basically sleeping. And that's what makes them harder to kill, especially because you rely on actively dividing cells for, especially cytal activity. So they're looking at TB from the lens of why TB treatment is so complex and long. Right, which is really because of persister/tolerant cells. So there's the initial rapid killing phase, but then there's always these residual mycobacterial cells that get left behind that are really difficult to eradicate. And I think what's cool about this paper is they show that persistence is not a single phenomenon, that depending on the type of antimicrobial you're looking at, the type of dormancy and the type of persistence actually looks different. It's not just, massive metabolic downregulation of the same genes every time. And I think, they do a lot of very detailed dissection of essentially this is with an in vivo study. So I think it's very cool because they're able to look at after 4 weeks of treatment of these mice, comparing rifampin, for example, to a completely other isoniazid, right? Like what do those dormant types look like? What are the cells remaining behind that are really difficult to eradicate look like on a phenotypic level, genotypic, transcriptional level? And ultimately, I think they're able to show that it varies. Different drugs create different complicated bacterial persister phenotype. So it does differ based on drug. And it also introduces a very interesting concept of maybe we should think about that when it comes to sequential treatment and combination therapy, both, that maybe we can take advantage of these, the fact that different drugs have different impacts on persister and dormant cells. An example is they're able to essentially hypothesize why rifampin, pyrazinamide, and betaculin, the sterilizing trio, potentially work so well. So the general implications are, rifampin forces a much deeper dormancy. So there's levels of dormancy. So it really puts them to sleep. And then pyrazinamide comes in after, damages the membrane, which forces them to repair themselves. And they can't simultaneously be asleep and repair themselves. So that's essentially why that particular trio works better than another triple combination. So I think it's really pushing us to think deeper about mechanisms of action too.
Yeah, very, very, very cool, very interesting. We could probably talk for days on the differences of what is tolerance versus persistence versus heteroresistance versus dormancy. Actually, I have probably a couple of podcasts already trying to discuss this, and it's very difficult to really pinpoint that as a characteristic in a particular organism. I'm very happy that you have highlighted that complexity that varies with the drug bug combination, with the timing, with the type of infection. And bacteria are highly evolved organisms, and they are going to respond to this. And this is nothing new. We just are having the tools to understand to the granularity of this. So great. And this is a great example how you can dig deep in this phenomenon and add to the complexity of this. So great. So you have the honor to finish the podcast, Madison, with the last paper from JID.
All right. And so I cheated a little bit. So this is actually like two papers in one.
Okay.
And the reason for that is the first paper is a perspective that really changed the way I kind of think of infectious diseases, or at least let me look forward in a new way. So there's a perspective called The Changing Paradigm in Infectious Diseases, Host-Directed Medicine, and Implications for the Next Generation of ID Physicians. And it really talks about how we're in the era of manipulating the immune system, and when is that going to kind of trickle into the infectious disease world, and how that's going to potentially change how we treat infections moving forward. And so I really loved that article. I thought it was a great way, kind of like expand your horizons paper and think about things in a new light. And so I wanted to look for a paper in JID that kind of highlighted that. And so the research article that I couple with that is titled Triggering Toll-like Receptor 5 Signaling During Pneumococcal Superinfection Prevents Selection of Antimicrobial Resistance. So this paper used a murine post-influenza strep pneumo superinfection model to look at how do antibiotics alone and antibiotics in combination with immune augmentation alter not only treatment outcomes, but also can shift the populations of resistant and sensitive strains in an infection. So they are taking mice and they treat them with influenza, and then a week later inoculate their respiratory tract with either amoxicillin sensitive strep pneumo or amoxicillin resistant. And then they treat with either amoxicillin or amoxicillin plus inhaled flagellin as like a immune booster. And then they look at how are the bacteria killed. And so when you add the inhaled flagellin, you see that there is improved killing both in the ampicillin and the amoxicillin susceptible isolates, but also in the amoxicillin resistant one as well. And then they did a kind of competition assay where they mixed the sensitive strains 1,000 to 1 of the resistant strains. So the resistant is a really small subpopulation in the infection, and then treated with amoxicillin and amoxicillin plus the inhaled booster. And you see that when you just treat with amoxicillin, you rapidly kill off all of the sensitive, and then the resistant ones, outgrow. However, if you treat with both, you see that to be really suppressed in this model. So I thought it was an interesting way of looking at host-directed therapies and antimicrobial resistance, and how, maybe in the future we'll be really probably dialing our immune systems as well as our antibiotic choices to help treat complicated infections.
Yeah, this is a very interesting concept that I still need to wrap my head around it, and how, because the immune system is so complex, and we see now in cancer how we manipulate in this for cancer, and there are very success stories. We know, at least from the infectious disease point of view, when the immune system goes sort of out of the way, or not very well controlled with these therapies, these things are really devastating in patients.
Yep.
But there's many attempts in intracellular bacteria, particularly with salmonella to try to understand these possibilities. Time will tell, but I think this is a very interesting concept that definitely you kill better, you decrease the ability of bacteria to develop resistance and the chances that they have to do it. Well, this has been an amazing conversation. We had a fantastic discussions on IMARI. I remember next year in January, at the end of January, we have in 2027 IMARI again. Hopefully, we'll also discuss a new session with new papers. Research is still very active in both societies, and I'm so happy to have this collaboration between the two journals, bringing top papers and top research to the front fold. So I want to thank you guys for taking the time to read these papers, to analyze them, and think about this. And I really appreciate that. So this is Cesar Arias, editor-in-chief of AAC, signing off.